So here we are to Unit 7 in Animal Science, which covers genetics. And I know the slide set only says 22 slides, but don't let that mistake you for an unimportant unit or one that's going to be probably right up there in one of the more difficult ones that we talk about, because genetics are always difficult to understand for some people and how to select and manipulate and apply those genetic principles. So this is chapter 12 in your textbook, and hopefully you've dug into that reading, that 18 pages of critical stuff right there about the middle. Content here, the first concept that we'll talk about is the process of gametogenesis, or making those gametes or sex cells that occurs in the male and the female. When we talk the sperm, that's spermatogenesis, and the female, that's oogenesis. And cell division on those two occurs by the process of meiosis. We will go through that process in class, and I would make the assumption that many of you have already talked a little bit in high school biology about the processes of meiosis 1 and 2, and drawing and diagramming what happens in each one of those. We'll revisit that in the classroom discussion time, but as we go through step by step, and remember that other than interphase, the processes of prophase through cytokinesis occur twice in meiosis. So interphase is the resting phase. That you see that used in textbooks, that term used a lot, and this is maybe the most important part of meiosis, because that's where you actually see the chromosomes duplicate. And so we make an exact copy, send it through the copy machine, so to speak, to make an exact copy of the entire genetic profile, because we're going to divide that here pretty quickly. In prophase, then you'll see the nuclear envelope begin to disappear, and you'll start to see some centrioles appear, the poles, so to speak, of the cell begin to appear, and those DNA strands are now tightened, wound down, and you begin to see them under the microscope. In metaphase, you'll begin to see the chromosomes very easily now line up on the equator or metaphase plate of that dividing cell before we get ready to pull them apart. Anaphase then is the first division phase of meiosis, where you'll see the spindle fibers reach in and grab each piece of that genetic profile, each chromosome, and pull it in half, and pull it towards each pole of the cell. First time that you see an active division in meiosis. Lastly is telophase. You'll see the full division, then the full spreading of the chromosomes, pull in half towards each centromere. You got half a copy on one side, half a copy on the other side, or one copy on one side, one copy on the other, because remember we started with 2. And then cytokinesis is the active pinching off of each one of those now brand new daughter cells. We're going to take that one now. Remember, both of those will go through meiosis 2. We'll take that one and go back through prophase, metaphase, anaphase, and telophase again before we have cytokinesis a last time. And now we'll have a half a copy of your genetic code. Again, we'll revisit this diagrammatically in class, but the result 1 and 2 in spermatogenesis, we get 4 sperm cells from each primary spermatocyte. When you talk about oogenesis, we produce, after the first meiotic division, we produce one large oocyte and one smaller one, which we call a polar body. The polar body does not go through further division. It is resorbed. We try and have one very good, very large, very healthy oocyte. The second division then occurs, so we get again down to that half a copy of genetic material. The second meiotic division occurs, so again we have one egg and a second polar body. So that one very large ovum and a second polar body. That second polar body also is resorbed. It's reabsorbed into the ovary. So, and that's the start. If we're going backwards into the previous unit that we talked about in reproduction, that's the start. Here's now where your egg is made to go into that developing follicle. Here's some diagrams that are kind of explaining what I was just talking about earlier, where we have a primary germ cell, a primary oocyte, and a primary spermatocyte, and where we get the resulting 4 sperm and 1 egg. Fertilization, then, each sperm and egg provides one chromosome to each pair. The fertilized egg, once that happens, we call that thing a zygote. Zygotes are diploid. The gametes are haploid, which is one copy of each gene. Diploid is 2 copies of each gene. All these events are always random. There's absolutely zero way right now on a commercial basis in animal agriculture to guarantee which mating that you get. The good thing is this provides a lot of genetic diversity into animal agriculture to our animals. The bad thing is sometimes we can't control what that genetic diversity is, and so we can have things like genetic abnormalities. We can have things like genes that aren't necessarily helpful for us in making our product or making it efficiently even. So, but it's important to remember that each fertilization event is completely random. We have no idea which sperm carrying which piece of genetic code is going to mate with which egg and what parts of the genetic code for mama that egg is going to be carrying. So it's always random selection that way. Here's an example, a diagram. Sperm and egg fertilization occurs, and then we have that developing zygote. Note that each sperm and egg has a half a copy of the genetic code, and when they unite, then we again have a full complement of DNA genetic material. The DNA, again, remember that genetic code, deoxyribonucleic acid. It's a double helical structure, so imagine a ladder that you have at home, and you take that ladder that has the crossbars on it, and you just twist it a few times. And those rungs don't ever break in this kind of rubber ladder looking thing, that double helical structure. We send that through a copier and make a copy, and that's where each of the genes are located whenever we divide those. And each chromosome location of each gene on a chromosome is called a locus. So here we have a gene pairs, and these chromosomes would be paired up before we split them, and you would have genes, let's say, for growth, fatness, hair color, again fatness on the bottom, growth, and horns. So a couple different things going on, and as a fertilization and division occurred, you would have no idea whether you got something beneficial or not, or what hair color you actually see it. Genes and chromosomes are paired are homologous. They transmit of traits to offspring. Again, it's entirely dependent upon which chromosomes are passed on and what fertilization we have. Sex chromosomes, remember, are X and Y. The male carries both. The female can only contribute an X chromosome. So a female can only contribute a female egg. The male contributes either a female sperm or a male sperm, depending on which one. Homologous chromosomes may differ in the way that they influence a trait. If they influence the trait the same way, we call that homozygous. So if we have for example, an Angus lots of times throws dark calves. And so clearly in that instance, most of the time we'll say that Angus are homozygous dominant for black hair color. No matter which chromosome they pass on, it almost always is the same black hair color, or B chromosome. You may have instances where we have what we call heterozygous, where they have a different effect on the trait. So think shorthorn cattle here, where we can have red cattle, which is the dominant trait, or white cattle, which is the recessive trait. And you can actually have what we call a roan, which is incomplete dominance, and we'll talk about that later. Each of these traits are called alleles, and we'll get into doing some crossing in a couple slides, and then we'll certainly practice it more in class. But generally speaking, one gene is always dominant and the other, or suppressed gene, is always recessive. And there's some different gene relationships, again, as we get into more advanced genetics, you can think about, but generally just keep it simple here: dominant versus recessive. We can also talk about the concepts of genotype versus phenotype, and they're very simple. Try not to get them confused, but genotype is the genetic code. Phenotype is the expression of that genetic code. So phenotype is what you see. Genotype is the DNA code. Again, we'll practice some of these matings in class, and so they're just listed here, some of the basic things that we're going to do, some mating practice on using the Punnett square. And if you've not done very much of this, we'll catch you up and we'll go through some activities in class that hopefully you'll have a fairly decent idea of how this works by the end of the year. Here's an example that your textbook supplies, and each one of those B's at the top, you see the big bolded B's on the top and the big B and the little b on the left. Each one of those are the alleles that I'm talking about. So if we pretend that the male is the two big B's on the top and the female is the big B and the little b on the left, the homozygous dominant genotype on the top, the heterozygous genotype on the left. And so if we mate them so each parent contributes that gene, that allele, and so we have two that would have the genotype big B, and two that would have the heterozygous genotype big B, little b. And if we have complete dominance here, all of the phenotypes, all the calves are going to turn out black. But again, we have a homozygous dominant and a heterozygous genotype. Here's another example where we have two heterozygous genotypes. Let's again pretend that the male is on the top and the female on the left side. We do our mating, so we have one big B genotype, two heterozygous big B genotypes, and one little b genotype. Again, if big B or black is a completely dominant trait, then we're going to have 75% black offspring and 25% red offspring in a 25% chance of having a red calf. So keep that in mind. And we have 25% homozygous dominant genotype, 50% chance of heterozygous genotype, and 25% chance homozygous recessive genotype. Class. Gene interaction, I mentioned we have some different things that may go on. Dominance interaction exists when the dominant trait always suppresses or covers up the recessive trait. So if you always have a black phenotype that covers up a red phenotype if the black is present, then that's a dominance interaction. We also have that relationship called incomplete dominance, where there's a third expressed phenotype that's different from either homozygous dominant or homozygous recessive genotype. So that would be, for example, the red, white, roan phenotypes that you might see. Again, if you're thinking, oh my gosh, this is difficult stuff, we're going to go through some of this in class. The interesting thing about some of those heterozygotes, those third phenotypic expressions, is occasionally we find things that heterozygous genotype actually is superior to the homozygous dominant or homozygous recessive animal. We call that hybrid vigor. That's especially prominent when we talk about the concept of crossbreeding. That's why you see so much crossbreeding in the beef industry and in the pig industry. You see a lot of pigs that go to market, they're all the same color. They're all pink. That's the result of generations of crossbreeding that result in the animal that's genetically the most superior for meat production. So that's an example of hybrid vigor, where you take the benefit of one breed and complement it with the benefit of another breed, and creates a genetic outcome that's superior in performance and superior sometimes even in genetics than either of the purebred parents. So that's what we call hybrid vigor, or heterosis is another term for that. So you see the terms of complete dominance, lack of dominance, or even over or codominance that go along with gene. So here's sometimes what happens is when you see different genotypes, when you have complete dominance, what occurs? When you have lack of dominance, so you could have an equal expression of all genotypes, and then over dominance or incomplete dominance, where the heterozygote genotype is the best performing one. Now, keep in mind, when we talk genetics and then add environmental impact, that the environment has a huge influence on the expression of the genetics that we've bred. So you can breed the absolute perfect, genetically perfect animal out there. And if you have a crappy environment, crappy nutrition, terrible facilities, terrible management, terrible heat stress, terrible health problems, the genetics cannot really help you that much. They can be hidden. So, and likewise, we can have animals in really poor environments that have great genetics, and we take them out of that environment, put them in a better one, and all of a sudden those animals flourish. So always keep in mind, genetically, there's oftentimes more opportunity, there's more production capability there than what we're maximizing in animal agriculture. As I say, give an example here, and I'll just give you one real quickly, and then we can talk about a few more in class if you like. Let's say, for example, that we've crossbred the absolute best performing steer. We've complemented the correct breeds. They have the absolute ideal carcass merit. They're very, very fast and efficient gainers. They're very good in terms of frame size. They're exactly what we're looking for. They have the genetic potential to grade out prime. We're looking at some awesome steers that are going to make great steaks to throw on the grill. And we take those genetically superior animals and we throw them into the feedlot, in which we don't have very much concrete at the feeding rail, if any at all, and we have an environment that's very rainy with poor drainage, and so those steers are trudging around in mud up to their bellies. Now, do you think that those animals are going to perform to their highest genetic potential? Very, very likely not. Now, will they perform better than a poor genetic animal? Maybe. But they will never reach full production capacity because we've provided them and put them in an environment in which it inhibits full expression of their genome. So keep in mind, that's an issue out there in animal agriculture, that probably our genetics are far surpassing what we're getting out of them in almost every facet. And that's something that depending on what part of animal agriculture you like, might be a spot for you to investigate a potential career opportunity. We can talk about some more examples in discussion. Some biotechnology things coming along, and we've talked about a few of these in terms of embryo transfer, sexing semen, superovulation. Add to that the concept of cloning, which has been used in a variety of instances. For example, cloning started with Dolly the sheep. If you read your history books, you can go out and Google that and check out Dolly the sheep if you've never heard that, the very first cloned sheep. We've used the technology somewhat in the dairy business. Go out in there and finding those high-end genetic animals, those best show cows, and we're cloning those, taking some hair snips or some ear punches and taking those cells to the laboratory and cloning that animal. It's interesting to note that not all of the clones turn out to be as exact matches to the parent as what you think. There's almost always the miracle of genetics happens a little bit differently. There's a slight different color marking, or again, environmentally we have some impact. So cloning is part of the issue out there. The things that you see towards the bottom with regard to genetic markers, gene therapy, and gene selection are coming along. We're able to now mark and identify specific genes within the genome that maybe cause a genetic abnormality, that maybe we can splice in a different gene and use gene therapy to eliminate a genetic abnormality or disease out there. Interesting that those things are continuing to develop, that we can maybe eventually call use through the process of gene selection a designer calf or a designer horse or a designer pig, that we can go through more or less a catalog and select which genes will be part of the mating and how they will be selected. I think that's generations away in terms of human generations, but certainly it's on the cusp of happening. I think the technology is coming along and probably is there as we speak. So when we talk about applications, there's a lot of different things that we can utilize. Another example here on how genes can be used or modified or duplicated is we took that bovine somatotropin and we were able to duplicate that in the laboratory, inject it back into cows, and actually raise their capability to make milk. That's one example. We can take genes and harvest a gene from one species, enhance it or alter it, and insert it into another species. That's called transgenesis. We can take nuclei from one animal or one individual and insert it into another nucleus. That's called nuclear fusion. We have, again, a ton of different things coming along that provide us with the opportunity to maybe do some different things genetically. some of the question is this what we should be doing? Here comes a moral bit for you to think about. It is, should we be doing some of these? Because if it begins occurring in animals, now you get to the question of should it be occurring in humans? Because again, like I said, I believe the technology is already there. Just is it commercially available and affordable to be used in animal agriculture? And people will say, should we be doing that? Good question. Good question. Here's an example of a fertilized swine egg that's being injected with genetic material here in a vacuum. They put the vacuum on the left and insert the needle on from the right side of your screen. Trying to figure out, can we do something different with this pig egg? Some take-home messages, and obviously we've barely scratched the surface over the last 20 minutes of genetics, and obviously I've not given you a very great idea on how to do some of these crossing, which we'll take care of some of that in discussion section and reinforce it with some activities and lab time. But I would stress to you, your knowledge of genetics is really key to making improvements in your herd. This should be coupled along with the reproductive talk that we had in the last unit. You should try and dive into genetic potential. I will encourage you to think about this. Certainly some of you purebred breeders out there that are in my class are going to be saying, yeah, that's something that I'm really interested in. And my commercial folks in class are saying, I don't really care about that. I want a cow that makes a lot of milk, a steer that finishes very efficiently and grades out well for me, or a pig that has great carcass merit. And I echo those sentiments for you commercial guys, but I will tell you that a lot of your genetics are coming from the purebred side. Pay attention and also get into the discussion of what kind of genetics are profitable for you on the commercial farm, whether it's the commercial feedlot, the commercial dairy, the commercial hog farm. Dig into those genetics and know what is available to you that can maximize profitability, and get involved in those breed industry things and discussions on where the future genetics of each animal industry should be going. It's important to you. So get plugged into that, even if you're not interested in showing anything or raising any bulls or doing any of that. But get into it's profitable for you to understand it. Be able to connect and predict gene interaction and potential outcomes. That helps your mating success when you're making those decisions. And then understand always that high genetics and investing a lot of money in high-end genetics is not going to overcome poor managerial choices. Great genetics won't overcome any of that. So you still have to do your part as a manager or as a consultant of managers, or some kind of in the service industry person. Help your producers along, improve their managerial skills, improve the environment for the animals so that we can maximize genetic output. So hopefully those are some good take-home messages. I assume we should have some very, very robust and frank discussions involving genetics in class, and I'm looking forward to that discussion. Thanks for listening.